So we will start from questions from the audience, and then this will maybe stimulate further discussion between Aaron and Mike.
Okay, let's do like that. So we open the floor for the audience, so please ask questions. And I see Susan.
Hi.
Maybe we give the first question to Susan as female.
Susan, you're muted.
I don't think so.
You're okay.
Okay now? Okay.
Yes.
Yeah, we can hear.
Yeah, great, thanks. Two fascinating talks. My mind is completely blown by them. Thanks, both of you. What I'm wondering now is, having heard Aaron's lovely thing about virtual machines having to be in place to build the next level of virtual machines and so on, and now also having heard about Michael's bioelectrical networks at the base level. Is the bioelectrical network something like the almost the machine code of the base layer on which we could then build Aaron's virtual machines at higher and higher levels? And so have we now seen the base level, and can we go on and build the higher levels?
I think that's a very important point. So Chris Fields, with whom I've written some things, has is thinking exactly on these lines about multiple virtual machines implemented in part by bioelectrics and by other things. One comment I'll make about the machine code is this: and this is when I give a talk to regenerative medicine audiences, I sort of go through the history of computer science, and I have this great picture of somebody operating a computer in the early 50s, and she's sitting there and she's literally moving the wires around, right? So it has to be at that point it had to be at the hardware level. And so that's where all of biology, biomedicine, let's say, is today, is very focused on pathways, genes, right down at the lowest hardware level. And so I'm making the argument that in complement to that, what the bioelectric interface allows you to do is to operate at a much higher level interface. Because what we can do in what the bioelectricals, for example, people often say to me, can you use these bioelectrical states to get stem cells to become, I don't know, neurons or something? But possibly, but that's not what the bioelectrical layer is for. It doesn't specify microstates. What it specifies is make an eye, or your eye should be this big, or your leg should be over here, and it should have this many fingers. So it's a very large scale, high level descriptor that right. And so I think by operating at that level, we can do a lot better than machine code, basically, because and I think that's how evolution sort of discovered this strategy, right? That not everything is efficiently done from the bottom up as micro level.
Okay, thank you. Yes, please, Martin.
Yeah, I would like to ask, because I can see here step from like explaining morphogenesis through patterns of bioelectric circuits. But now the question is, what is this genesis of the patterns of bioelectric circuits? How this pattern, because if it was just accidental, then we would have very different individuals, different frogs or different.
Sure.
What makes one frog similar to the other frog?
Yeah, no, it's absolutely not accidental. So what happens is the genome of any given creature specifies the properties of the ion channels that those cells have. And those ion channels, in turn, constrain the electrical activity that is going to occur when you turn on the juice. So as with my students, I sort of say, look, imagine you have a collection of parts, and it happens to specify a calculator, and so you turn on the juice, and it has some default behavior, right? This is what that electrical circuit does, and that's just and what evolution takes care of is making sure that the properties are such that symmetry breaking, amplification, and self-organization is quite reliable. This is why every time it's almost certainly to be correct. However, what you then discover is that it's not a cheap calculator, it's a reprogrammable calculator. So that, yes, it has a default behavior. And when you turn on the juice, it always says 0, and it starts off like that, and that's nice. But actually, then you find out that with experiences, not by rewiring the thing, but by pushing buttons, by giving it signals, you can actually get it to do lots of different things that maybe normally it doesn't do. So I think what evolution takes care of is making sure that the hardware of the cell, specifically the ion channel driven networks, have a default behavior that with no other perturbations, they always do this. And in fact, with perturbations, they might still do that. In fact, as I showed you, they are very robust to these things. However, once you understand how it works and you found the API, so to speak, right, you found what signals actually make this system do various things, then you can really take advantage and take it off course into some other direction. But the evolution makes sure that the default behavior is adaptive and stable.
Thank you very much.
Okay, I think the next question is Susan again, and then Pedro.
Oh, sorry, I must have kept my hand up. I will take my hands down.
Oh, sorry, Pedro. Pedro, please.
Okay, I have two very brief questions. Well, it was very impressive what you have been talking about. Have you explored with different types of cells, tissues, and species? Do you find that there is some more creative kind of tissue in these experiments? And the second question, what's the relationship with this sort of bioelectric code, with the force field code?
Okay, the first question, various different types of tissues. So people, my lab and other people in the field have done everything from bacterial colonies to human cells, and all of them. This is a very highly conserved system. It's all tissues and organs participate. Some are more or less tractable. So, for example, heart cells are difficult to work with bioelectrically because they have a very clear idea of what voltage they want to be. They want to beat at a very specific rate, and it's very kind of, you have to really fight to get them to change, which you might want to do. We've done regenerative applications where you can we electrically try to grow new heart tissue and so on, but it's quite difficult actually, because they're very fixed in their electrical rhythm. They don't want to change. But most cells are much easier than that, and they will change reliably. And this is true. We see this from bacteria to humans and so on. With respect to your second question, I'm not sure I understand. What force field code do you mean? Biomechanics or something different?
There are different ways of mechanically interfere with gene expression.
Biomechanics, yeah. Got it. Yeah, understood. They absolutely do interrelate. I mean, I told you a whole story about bioelectricity. Someday someone will tell a similar story with biomechanics, probably. They relate in two ways. So there are stretch activated ion channels, so the biomechanics can control certain electrical properties, and the reverse is true too. So there are proteins that under electrical voltage potential can push and pull and exert force. So there are certainly, evolution has certainly found adapters to move between the biomechanical computations and bioelectrical computations. And so I think these things work together, but we don't happen to study much the mechanics.
We have Aaron.
Right. One of the people who was here for the first 2 hours, but I guess he's had to go, is a well-known physicist whom I first met when we were both students, and we've intermittently kept in touch. I will try to communicate with him about the question that's in my mind about, but it may be one that somebody else here would be able to respond to. It is this, that the processes that go on inside the egg, and also in all the things that Mike was talking about, seem to involve very fundamental physics, but extremely low energy physics. I mean, there's nothing like all the stuff that goes on where they have to build these things in Switzerland and whatever, high-speed smashing things together. By contrast, it's enormously peaceful, but it's enormously productive and rich and complex in its structures. And in my mind, there's a question whether there are implications here for fundamental physics that no physicist has ever thought about because they don't think enough about biology. I'm thinking in particular of a talk I heard by a distinguished physicist who's head of a physics center. It was recorded maybe in the last year or so, which is called The Astonishing Simplicity of Everything. That's the title of his lecture. And he was claiming that everything can be reduced to one rather complex and abstract equation. But it's not at all obvious to me that the material that was being presented by him and by other physicists that I've heard talking can actually explain what's going on in the things that Mike was talking about and that I was speculating and waving my hands about, but I think are related to what Mike was talking about in quite detailed ways potentially. So I'm just wondering whether some of this could be potentially the source of a new revolution in physics. I don't know if anyone here knows enough physics to tell me that I'm talking rubbish or the opposite.
Well, the only thing I would say about that, and I think it's a great question, but I take a little bit of issue with the characterization of how peaceful it all is, because if you scale down, right, if you scale down to the level of a cell membrane, a depolarization event is basically like lightning. It's got the proportional the electric field like a lightning strike. I mean, but to the actual cell, a depolarization event is a massive it's a massive thing. But I completely agree with you that, for example, these VATPase pumps are functioning right at the peak of efficiency, the thermodynamically allowable efficiency, which we don't even get close with our machines. So, yeah, I mean, I think you're right.
Yeah, I must remember to make the qualification that you've just made, but the point still remains that the total amount of energy involved in the production of a chick compared with anything produced by any human factory is just minuscule.
Thank you. I think Martin has a question.
Susan was first. Susan?
Susan, please.
Oh, right. Yes, this is real this time. Sorry about last time. Yeah, so again, I'm trying to reconcile these two fantastic talks, and Michael was talking a lot about this morpho space or whatever, and a lot of it about structure, and Aaron was talking a lot about behavior, like the avocet chicks that can behave straight away. So is this morpho space also able to capture that transition from just structure onto complex behaviors as well? Is it kind of the same source as being able to explain why Aaron's chicks can forage right from the get-go?
Yeah, so the one thing I want to say about that is that I'm suggesting a peculiar a mental switch here, which is to not think about, I don't think that anatomical morphospaces are about structure at all. I think that anatomical morphospace is behavior. It is the control of clever behavior. It is exactly the sort of thing that Aaron was talking about in terms of, yeah, you start as one cell, you need to know how to construct a salamander. That is an incredibly complex behavior. It's just that it's behavior that takes place in a different space. You also need to know how to manage your physiology in terms of, hey, what happens when there's too much potassium and not enough chloride? And you also have to know how to walk in transcriptional space, which genes come on and off at certain rates. So to me, all of this is behavior. I want to, and I realize this is a controversial notion, but I propose to generalize this idea of behavior away from the familiar three-dimensional space that we see brainy animals running around in, and say that behavior is sometimes intelligent problem solving in lots of different spaces that we're not really primed to see, right? Our cognitive apparatus is really focused on 3D behavior, and that kind of thing. But I absolutely think that morphospace is exactly the behavior, and it's exactly the mysteries that we have are exactly the mysteries that Aaron pointed out. That's how we started this whole thing. How does the thing know what a correct salamander looks like? I mean, it's born. Where does that come from? Exactly the same problems.
So this other space might be the linking between the complex geometry going on in the head So the geometry is coming from the outside 3D space, but that complex behavior in the head is in a morpho space. Is that?
I think that's true. I think a lot of the things we see possibly, I mean, I don't have the background that Aaron was talking about, but would be recognizable by him as geometric constructions. You know, these tissues know how to, they understand things like, hey, I'm going to wrap around. I'm a nerve and I see some bone, so I know what to do. I'm going to wrap around like this, right? They do these constructions all the time, and I think it's exactly the same mystery here.
Now, I would like to ask about time dimension. When we look at morphogenesis and these processes, it looks like there's huge number of operations. So it is very fast in some sense. Fast in the sense of number of operations, what is happening in time, how many parts are involved in this. When we look at, for instance, work of neurons are working like a little bit more global scale, but transmission of signal is very slow. Yeah, it is like incomparably slower than, for instance, electric current. So can we explain this? Because for me, original question is, okay, why our nerve system, for instance, which is controlling, is so slow? Can we explain it through some kind of relationship with morphogenesis? Maybe I will explain it because maybe my question is not precise enough. For instance, in neurogenesis, like when we have human embryo, there are like million connections between neurons are built within 1 second. Yeah, so it is very fast process, but transmission of signal in the neuron is very slow. It is something like about 300 meters per second. How can we explain in some way why we did not develop system of control through neurons by neurons, which would be faster? And can it be related to some processes in morphogenesis?
I have a mischievous answer, which is that the neurons aren't really doing the things that we attribute to them in all of the psychology and neuroscience labs. They're doing something else, and the real work is going on at the level that Mike and I have been talking about, not visibly and not so easily detectable. And the neurons are doing something else, which is providing infrastructure and backup mechanisms and distributing resources and whatever. But it's mischievous, because if it's right, then there's a huge amount of stuff in print and that's completely wrong.
Yeah. So my answer would be, we usually tend to think of it the other way around, which is that, you know, developmental decisions take days. You know, the basic morphogenetic machinery is actually very slow. Deciding if you're going to be a head or a tail or something like this, it might take 24 hours to make that decision. The developmental bioelectricity changes very slowly compared to neuronal electricity. And in fact, I think what evolution did when behavior became important and animals started chasing each other around to try to eat each other, speed became of the essence. And then you have all these innovations like myelination and all this stuff that actually operates at a much faster scale than developmental events. And in fact, we use specifically the reagents that we use, for example, to track voltage changes. We can't use the same dyes that neuroscience uses because they're tuned to be much faster. They're all rapid response dyes and things like this. We have to use different reagents that average over all of the tiny fluctuations and report changes on the scale of hours. You know, our imaging so one of the things I often have my students do is as a starting exercise is I say, look, take any paper in the field of neuroscience, go into put it in Microsoft Word and do a find replace. And everywhere that it says millisecond, put in hour, and every place where it says neuron, put cell. And that's it. And now read the paper and see what you got. And usually everything works nicely, and you get all kinds of interesting hypotheses and so on. But things that take milliseconds for the nervous system is minutes or hours in our space, and all of our experiments have that much longer time scale. We don't
because
collect.
I was thinking about Aaron's starting point about this chicken, which is in 20 days develops from very simple system to something extremely complicated as a chicken, which when it's hatched is walking and picking food and so on. So from this point of view, it is like huge amount of processes, but of course it could be that these processes are parallel. That's
Well, they are.
This case.
Yeah, and it's a little bit apples and oranges, but you can also ask in 21 days how much computation could a nervous system have done in 21 days, right? That's depending on how you measure these things. 21 days is a long time to compute things on the scale of neurons that are doing things in a millisecond kind of essential scale. But I think you're right in that it's very much scale dependent and observer dependent. It's what level are you interfacing with the system, right?
Thank you.
Can I just make a last remark before we leave this point, which is that one of the things that wasn't in Mike's talk in the same sort of way as it was in mine, although there were hints of it, is that I was talking about the provision of information during the development in the egg that was going to enable the chick to do all kinds of things in the environment about which there is no information inside the egg. And therefore, there must somehow have been a process by which the history of the genome got information into a form that could then use this mechanism to produce ready forms of intelligence that are available, as you saw in the chicks. And of course, I think much more subtle things to do with things like language development and others, but mainly the immediate behaviors which just don't have to be learned. It means that what's going on is not just assembly of all the parts and setting up all the connections and production of all the possibilities for sensing and moving. It's also abilities to solve problems that they could not have known about and worked out the solution to on their own. The solutions had to be worked out by the evolutionary history.
And actually, that's a super interesting point. I'll add one thing to that about solving problems you've never seen before. We had a paper recently where we showed that if you take planaria and you put them in a solution of barium, so just barium. One of the things that barium does is it blocks all potassium channels. And so that's a major problem for cells if they can't pass potassium. And what happens is to these planarias, their heads explode. Overnight, literally, their heads explode. But okay, and that's not too surprising, toxic and so on. But the amazing thing is that if you now take these planaria and you keep them in barium for another couple of weeks, they eventually grow new heads that don't care whatsoever about barium. They're completely barium adaptive. And so we asked, well, the amazing thing is, of course, that planaria don't see barium in the wild. There's never been an evolutionary history of resisting barium, right? So what is going on here? So we did a very simple experiment, which is we did next-gen sequencing on normal heads versus barium-adapted heads to simply ask the question at a transcriptional level. Not that that's where the answer had to be, but we thought we would look. And just ask the question on a gene expression level, what is different between heads that explode in barium and heads that are completely barium adapted? And the answer is that out of all of the genes in the planarian genome, which are, 20, 30,000 genes probably, only a few dozen have been altered. The transcription, not the DNA, but the transcription is altered. And what the cell has figured out how to do is, out of this enormous space of all the possible combinations of genes it could have up and down regulated, it picked just a few dozen to solve its problem, a problem that it's never seen before. And this is, I have no idea. I'm not going to give you the solution to this because I have no idea what the solution is. What I imagine to myself is you're in a nuclear reactor control room, right? There's buttons everywhere. It's melting down. You have to figure out, you don't know how to run this thing. You have to know, and you don't have time to start flipping buttons at random, mostly because that'll likely kill you long before anything else happens. So you can't just turn on, there's no time for gradient descent. There's no time for selection because they don't, the cells don't turn over that fast. Somehow, in a very short amount of time, this thing walks through transcriptional space and identifies exactly the region that is going to protect it against this crazy physiological stressor that was blowing it up, a few days ago. How, what that is, I can't begin to tell you, but we have examples like this of amazing problem solving in these weird spaces that evolution did not directly prepare you for. So there you go.
Thank you very much. Now I see so many hands that I don't know who was the first one. Let's see, Luke.
Hi, my question is not totally fair because I'm a night owl and I missed the beginning of the talk. I'm actually in the Pacific time zone. I worked as an undergraduate on a project on the evolution of vision. And there might be an answer to your last point in that, at least a hint, is that the theorist whose theory I worked on proposed that there's not merely random variation at a genetic level. There may be principles that structure the kinds of variations that happen. And those higher order principles, he proposed a principle of adjacency. So if you imagine retinal cells, so we work basically from retinal cells onwards, and how they, if you look at what happens, there's some grouping that happens. So that's the result of evolution. What if there's a higher order principle that says, the kinds of change we want to see at a genetic level would be driven by a principle, let's say a principle of adjacency. What do you get? So now I was a psychologist with the AI, or psychology student then with, using AI methods. So I didn't really understand anything about the chemistry, but I did work with, there's a PhD in chemistry who was, biochemistry, who was looking at that. And he had suggested that, there's maybe an underestimation of the, what to expect from random variation, basically. And I know that, selfish gene theory and all that says, no, actually you can get there very quickly. But anyways, that was the hypothesis. So that might be a principle. So I was thinking more generally, my question was going to be to you, whether you had some principles in mind for guiding that might bias, in a sense, the variation that we see. And I think it really follows on your last point that you're saying, well, we don't really know what happens, how they walk through the space. So then I would say, well, maybe something like adjacency, a principle of adjacency. I can send you the paper after if you're interested. It was my honors thesis.
Yeah, please do send. Yeah, no, I think you're absolutely onto something. We've taken a lot of inspiration from work on retina and pre-processing in the retina and top-down control and vision and all that, because you're absolutely right. There are, for example, our work on brain repair was driven exactly by ideas of edge detection from retinal processing, because that's exactly what's going on in the system, just in a different space. So I think you're absolutely right. All kinds of things map on, like perceptual bistability. There's we have, it plays a role here. All kinds of concepts like that. I think it's an incredibly rich and fertile sort of source for those kinds of principles. Yeah, absolutely.
The next question comes from Mohan.
Hello, can you hear me?
Yes.
Okay. Let me see if I can post this question correctly. So I have been sort of listening to both these parts of Aaron's talk I've seen before, because I have the luxury of being in the same institution as Aaron. And I heard Mike Levin's talk, and there are these interesting examples of these behaviors that are truly astonishing. So, and sort of building on what was asked by, I think, some other people here. So given that all the observations of these being low energy transactions and other things like that, is there any sense of there being some fundamental principles driving these kind of behaviors, or are we far from there right now? Do we know anything about what kind of processes come together to produce, I mean, some kind of cause and effect, or some kind of like, input-output mappings? Do we understand any of these at a level at which we can make some claims before we run some experiments? Not based on observations after? This is for either one of you. Yeah.
Aaron, do you want to take it first?
Okay, well, there was a part of my talk which was actually an answer to that question at a high level of abstraction, namely where I talked about the work done with Jackie Chappell, the biologist, on what we call the metaconfigured genome. And Susan Stepney published our first paper on that in a journal she edited. The idea there is that the process of evolution doesn't just produce things that start from scratch and build up in each generation. It produces specifications for the next generation at different levels of abstraction, where the later levels of abstraction wait until stuff has been done by the earlier levels to collect information. And then that is used to instantiate the higher level specifications, which are abstract. This is most spectacularly obvious in human languages. We all have the same genes, and yet there are thousands of different languages which vary in detail at all sorts of levels, including varying between sign and spoken and written languages and so on. And I think the only way that could have happened would be by using this sort of mechanism that Jackie and I have been talking about, although it's still too vague to be a scientific theory, and I've been talking to various people about possible ways of extending it. But it does mean, for example, and by the way, this applies not only to things like language. There are many things that are delayed in gene expression, for instance, sexual maturity, things that are relevant to that, which will make use of what had been learned at an earlier stage when an animal becomes sexually mature, and it'll be instantiated for the environment they're in. For instance, for animals that might have to mate in different environments or whatever, and look after the children in different environments. But anyway, in language, it is most spectacular, but in the ability to take in cultural products that have only recently been developed and make use of them, I think also depends on this metaconfigured genome, so that the later stages of gene expression, dealing with more abstract problem solving and so on, can use not only what's come from the earlier genetic gene expression, but also stuff picked up from the environment on the way, which provides new resources that weren't available for the same high-level genes or late genes in previous generations. So our 6-year-olds are doing things that none of their ancestors could possibly do, although the ancestors had exactly the same genetic capabilities as our 6-year-olds. But that's a bit vague.
I mean, for my side, absolutely, we have lots of examples where, I mean, we have very, as I tried to point out, we have very quantitative computer models now of the bioelectrical signaling, which make lots of predictions, which we test all the time. And in fact, these are driving therapeutic applications. So we have spin-off companies and whatever based exactly on this. So just to give you the one example of one of my favorites from the brain repair, we built a computational model specifically to try to tell us what kind of bioelectric manipulation would solve a tricky brain defect. We made predictions. The predictions drove a choice of ion channel drugs. We tried it, and it actually worked, and it repaired the brain. I mean, we have lots of stories like that. But most certainly, the field now is to the point where we can make we do make predictions and then test them in experiment after that.
So if I could just maybe part of that question was how much of this is data driven, or how much of this is actually adding to theory? I mean, are you making observations and saying here is a model based on these observations and therefore it works on the same data again? Or can we sort of learn any kind of fundamental theories where you can now go to a newer related problem and still somehow say there are these fundamental rules?
Yeah, that's the latter. We in fact have relatively little data. We can't do anything like data fitting or machine learning on data sets for the same data because actually getting these voltage images and so on is extremely difficult and time consuming. We absolutely have basic new basic theory which we apply to new contexts all the time. So we take things that we learned in flatworms and we do them in human cells. We take things that we see in the frog tail and we try them in rat limbs. And we take a lot of theory from computational neuroscience, from various aspects of cognitive science, and we try them and we validate them in these models. It's not a case of using the same thing on the data that you've already gathered. It's not that at all. With the exception that very early on, I mean, one of the very early tests, this was now, I don't know, 15 years ago, we were able to, we saw what the bioelectric state gives rise to an eye, and we said, well, I wonder what would happen if you use that in a different part of the animal, and sure enough, you make an eye there. So that's an example of learning something quite specific and moving it. But generally speaking, it's a much bigger move than that.
Thank you. I might contact you for a particular reference because I want to see what these building blocks are. But I'll do that offline. Thanks. Yeah.
I think it's Susan with a question now.
Okay. Yes, so one of the things you said at the beginning of the talk, Michael, was about all these different levels of cognition, and you've been focusing very much at the cellular level and saying there's an analogy between ordinary cells and neurons. But then you had these different levels, and you were talking about tissues and whole organisms, and then societies. So. Okay, so what is the analog of the bioelectric state at the social level, and how can you use the same techniques to observe, predict, and intervene to change social behavior?
So the mechanisms, of course, are going to be a little bit different. So it doesn't have to be specifically bioelectrical, but the basic principles, as someone was just asking about, are the same. So I'll give you a simple example of something that we're doing. In my lab, we are training ant colonies, not individual ants. We're training colonies. So the question is, can you communicate with the collective intelligence of the colony? Training individual ants is easy, but the question is, can you actually communicate with the collective? And so there you have to do some clever experimental designs to make sure that you're not actually training individual ants. But this issue of binding together competent individual agents into a greater whole and then communicating with that whole, and where by communicating I mean predictively altering its behavior, right? That's, as an engineer, that's my version of communication. And so so that's just one example, and there are others. You can try all sorts of things. And by the way, so that's an example from the kind of the top of the hierarchy. I'll give you an example from the bottom of the hierarchy. The classic sort of paradigm of determinism and genetics is gene regulatory networks, right? There's no mystery. There's no which gene turns on which other gene, and this thing is just a dynamical system and all of that. So we looked at that and we said, okay, is that really true? What if we try to train them? And what we discovered was that, in fact, gene regulatory network models exhibit all kinds of about 6 different kinds of learning, including associative learning, and so the general principle is you take things that you learned in neuroscience at whatever level and look for them at every other level. And we've not been disappointed yet. And you can go up, you can go down. It's, they're all over the place.
Great, thanks. The thing about the ants reminded me of Hofstadter's Gödelsche Bach ideas there. Yeah, okay. Oh, that's wonderful. Thank you.
We have Marcin and Pedro, I believe. Please, Marcin. Marcin, you are muted.
I'm sorry. I wanted to ask a question, which will be not really what I think, but like devil's advocate question. Mike said that they are simulating these processes on the computer. Then the possible consequence of this will be to say, okay, this means we don't get anything new for computing. if we can successfully simulate processes of morphogenesis on computer, this means that basically nothing new comes. As I said, it's not what I believe, but it generated in me this question. I think that this question would be to both Michael and Aaron.
Aaron, do you want to go first?
If you have some clear thoughts you'd like to express, I'd love to hear it, or even not so clear.
Yeah, I mean, my thought is this: there is a huge difference between potentially. It's sort of like, you know, why are we not all working on Turing machines all day long, right? There's and why, you know, maybe we believe in a kind of Laplacian determinism that in theory, if you knew everything, you could calculate. But these are completely impractical sorts of things. And when I say, people say to me, oh, why are you studying this cognitive stuff? You know, just go down to the biochemistry. Everything is fundamentally underneath chemistry. You should just do that, and that's fine. I mean, look, even if we believe that underneath everything is chemistry, and underneath that, I don't know, do people want to, molecular biologists don't really want to talk about quantum foam, right? They don't really want to go all the way down. They kind of have a favorite level, it's biochemistry, and why don't we just do everything with biochemistry? My point is this: in theory, maybe it's true that if you had control over all the molecules and you had some sort of computational system bigger than the whole universe that you could use to compute where every molecule is going to go, maybe you could achieve the same power. But this is crazy. We're never gonna do that, anything useful that way in our lifetimes. And if you want regenerative medicine, you don't want to micromanage the transcriptional state of individual cells to make a limb to tell every single cell what kind of cell it's going to be. No, what you need is a high level signal that tells the system, hey, make a limb here. Right. And it's very, so as an engineer and a worker in regenerative medicine, I think the practicalities of it are huge. I'm really not impressed at all by the idea that theoretically everything could be, you know, sort of computed at the micro level. That's great, but you miss a lot of the power of this thing, and it's just not.
Because, you know, like, my question is more like coming. We have like a group interested in morphogenesis as some way of new way of computing.
Oh yeah, I see. Okay, no, absolutely there are, because the thing is, you know, when I say these things are computing, they're not computing the way that our current devices compute. These are nothing like the modern architectures that we all use. There are principles going on here that I think have lots to tell us, and in fact, we have some of this going on in the lab. I have a couple of folks in AI trying to make artificial intelligence platforms that are based on not neural networks or neural kinds of principles, but something completely different that you see from biology long before. So on that level, absolutely, there are useful things here. It's not the kind of computer that we know how to make already.
Thanks. I feel relieved. Your answer is like giving me positive feeling. Maybe Aaron, what do you think?
I have a comment on that, which may or may not make you happy. It relates to the debates I mentioned involving Kant and Hume and what neural nets cannot do, but I didn't say, and what current computers cannot do, but I should have, because I think there are kinds of search space that fall naturally into the kind of discrete combinatorics that get implemented on various kinds of digital computers, and there are many variants. But there are other kinds of search space that I tried to hint at with examples, like the example of a triangle, where I said if you draw a line going between two corners and then move the third corner on that line further away from that, the angle will always get smaller. Now, I do not believe that the process by which human brains find that obvious is based on anything remotely like logic-based inference. It is, instead of logic and algebra make use of searches in discrete spaces. And there are ways of doing very powerful and complicated things in discrete spaces. But I think there are things that evolution discovered and brains use that, and I think probably are implemented in chemistry for reasons that are hand waving, but nevertheless, that involve searching in continuous spaces and finding and doing exhaustive searches highly economically in a way that no neural net could possibly do. And you're doing something very different from going through a collection of descriptions in discrete symbols of the different bits of that space. And that that example is one of many. I mean, a child can see that if you've got two rings locked like that, there's no way that, even if you can move them that way, you can move them that way, you can turn them around. There's there's a collection of infinities there. And a child has a way of grasping that despite all those possibilities, you're never, without breaking one of the things, going to get them apart. I don't think the child knows how it does it. I don't think anyone, brain scientist, knows how the child does it either, but they can do it. They can't reflect on it in the way that a topologist can. And then modern topology tries to map those kinds of problems onto things that become more like the discrete spaces, where there's a lot of stuff going on in topology that would say things about that. But anyway, I think that long before modern topology and modern logic and modern algebra and so on, animal brains were doing reasoning about what is and isn't possible. And they vary. Some animals are pretty dumb. There's a video that's quite popular of a cat with a corn cob trying to get through a gate which has parallel bars. And the cat keeps going to the gate and this corn cob can't get through. And it tries and tries. And then at a later stage, it drops this corn cob and by accident picks up one end of it. And then when it pushes its head through, that rotates the corn cob and it goes through. Now, a squirrel or a magpie or many other animals would not be fooled, would not be held up the way that cat was, because they can think about spaces in a richer way than the cat, that particular cat, and I think most cats can. There are different species that have developed different kinds of abilities. And I'm suggesting that those abilities to reason about what's possible and impossible in continuous spaces is very important in animal intelligence and a great deal of ancient mathematical intelligence, and is not yet modeled by anything in current computer science, computer algebra, computer logic, or whatever. I think it needs some different kind of mechanism.
You actually made me happy. Thank you.
And
you actually made me happy. Yeah.
Oh, right. Yes, that's what you were hoping for. Right.
Thank you.
And I think Mark's work might point at some of those mechanisms, even though that may not be one of his goals right now.
Thank you.
Thank you. I think Pedro is waiting with a question for quite a long time. Please, Pedro.
No problem. Michael, I think talking about general principles of morphogenesis, that you have cooperated recently with Carl Flintstone on a variational principle of morphology, I think based on minimization of free energy. If I am not wrong, unfortunately, I have not read it yet because I could not download it. So maybe I can ask you for the paper, but I would like to hear from you. How do you see this principle as a very general, vague constraint applied to the masses of cells, or for every cell, it's a kind of compass that this individual cell has to read the compass and to try to accommodate?
Yeah, I mean, there's a lot to be said about that, and a lot more that we haven't done yet that we need to do, but I'll give you a very simple example of how we've used this. And this was in a recent paper with Chris Fields. Imagine, so this is a story about multicellularity. Okay, how do cells cooperate together? Imagine that you're a single cell in an uncertain environment, and if Friston is right, then one of the things you have as a basic life form is you have a drive to minimize surprise. That is, you have some way of learning from your environment, making predictions, and you are continuously updating your model about what the world is like, so that you're less surprised than you otherwise would be, right? And so this is, of course, highly adaptive. So if you are going to be minimizing surprise, one of the best things you can do is surround yourself with things that are less surprising. What is the least surprising thing? It's yourself. So you might want to do is keep copies of yourself around you so that you now have a much better ability to predict what's going to happen next. More practically, what it means is that you are building now an internal milieu, which is more buffered against vagaries of the outside world and things that could happen outside. And so now you've automatically, if there's now this drive to do this, you've automatically constructed a system, which we see all around biology, where there's a protected niche inside, right? Chris calls it the imperial model, because basically there's the stem cell inside, and then there are copies of it, which are basically frontline infantry. They have to deal with the outside world and all the complicated and nasty things that are going to happen, but you are much more protected inside. And so one of the kinds of models that you can make based on first-instance principles is a drive for multicellularity that's based entirely on prediction of physiological responses and things like this, right? So that's just a simple example of how to use that principle to make to make predictions. The thing that we did in development is to say that that whole error minimization process is exactly that basically collections of cells have expectations about who's my neighbor, how long does it take for me to signal to my neighbor, what am I feeling mechanically next to me, what am I feeling electrically next to me. And when those predictions are not met, you have two choices. You can update your model, or you can try to move and get them to move and do things to make the model be realistic. And I think real development is exactly a battle of those two forces. It's learning from the new situation that's happening because you have plasticity, but there is a lot of inertia for that, because what you really want to do is make sure that the situation is exactly what you expected it to be, right? So there's this like battle of these forces. So I think actually Carl's work has lots of interesting concepts to explore in development.
Thank you.
Gordana, I think Annette wanted.
Yeah, Annette. Yeah, please, Annette.
Unmute.
Thank you very much. I want to ask a question that is a little to the bottom side of the electromagnetic code, decrypting. Maybe we had one example about the higher level, the social level, and your example of the ant colony. And now my question is about the gene and the DNA. Do you have any hints that it could be possible that the electromagnetic field surrounding or emitted by a gene and a DNA molecule could be depending on the sequence of bases? Is there any relation between the sequence of the bases and the field which the DNA molecule emits? The question is pointing in the direction of change of not the sequence, but maybe the field properties by epigenetic manipulations like methylation. Thank you.
Yeah, entirely possible. This is not anything that we study because our work has no magnetic component. We are studying only static electrical differences across the membrane. But there are people who do work on that kind of a thing, and I think it's quite plausible. Everything from ultra-weak photon emission to and from DNA to other types of there are a few labs that are doing this, and I think it's plausible, but I'm not an expert on that part, so I don't know.
And what I said implied that something like that was going on in the processes of control inside the egg, but I have no idea how.
Okay, and do you know a key phrase how to search for this? Not the Montagnier papers and the things which have been a little bit critical in the way they have been published, but newer work.
Yeah, send me an email. Send me an email and I'll see what I can dig up.
Thank you.
And I will talk to my physicist friend who isn't with us here. He's a very distinguished theoretical physicist with a Nobel Prize and very clever. and maybe he will have some ideas. His first degree was in philosophy, and which is how we met before we got converted to physics.
Thank you.
Pedro, is it a new question or do you have your hand raised? Okay, that was from before.
Yeah, I want to thank everybody for a great discussion. I have to run in about 1 minute, but thank you so much. Great questions. Yeah, just a wonderful discussion.
Well, we think it was a really great lecture. I think everybody enjoyed it very much. It was fascinating, eye-opening experience.
Cool.
Yeah,
And not too
really
scary.
inspiring. We have something to think about from this meeting of two great minds. Thank you
At the
very
same
much.
time, I would like to thank Aaron for his lecture. Both lectures, actually, it was a great combination of these two lectures. It gave broader perspective on these issues. So I think that we really are lucky that we could have both Aaron and Mike here with us, and it is something I will remember for years.
Yeah.
Thank you so
Indeed.
much, everyone.
Thank
Thank
you.
you.
Thank you very much.
Definitely. Yes. Thank you.